Everyone, thanks so much for having me today. It's a big honor to be here. Thanks to Randy and everyone who's made this come true. And today's talk is going to be about artificial intelligence and how it relates to biology. And I'm also going to talk a little bit about ChatGPT, which many of you might have heard of. So a bit about me, I'm from sunny San Diego. I study computer science at Stanford, and I'm really excited about AI for the last couple of years. I've published several papers in the area of computational biology, and I've been an intern at UC San Diego in Scripps. I'm here on behalf of the RISE program, which is an amazing scholarship. And it's for people between the ages of 15 and 17. So I'm sure many people here are eligible and should definitely apply. And I tweet a lot about AI and research on my Twitter account. So if you're interested, my handle is it's Andrew Gao. So brief overview, I'm going to talk about my research on disease diagnosis with artificial intelligence, drug repurposing, generative AI, some other applications, and then the part that everyone's really scared about, AI safety, if anyone's seen Terminator. And there's a term called AGI, which is related to AI. It's artificial general intelligence, and it's like Terminator. And it's actually all around us already. In fact, it's behind me, AGI. So yeah, it's already here, guys. So first, I wanted to work on lung cancer diagnosis. It's the leading cause of cancer deaths, diagnosed at late stages. And the current way you diagnose it is with tissue biopsies, where they stick a needle into your lung and check for cancer, which is really painful and expensive. MicroRNAs in blood can be non-invasive biomarkers or clues to help diagnose lung cancer. And several studies out there have identified microRNAs that could be used to detect cancer and classify cancer. But the issue is reproducibility between studies. So has anyone heard of a little startup in the Bay Area called Theranos by any chance? Yeah, so things like that, where one research group claims they've got 99% accuracy for detecting cancer, another group can't replicate the findings at all. So with my research, I wanted to identify reliable, consistent biomarkers across various studies that have a biological relevance to lung cancer that can be used to detect this terrible disease. So I analyzed data from 4 different studies. Each study claimed that they had identified hundreds or thousands of great, accurate biomarkers. But when I did a little Venn diagram to find out how many of these microRNA biomarkers were identified by all studies, only 13 out of thousands were repeatedly identified as reliable biomarkers. So I took these 13 and I analyzed the fold change, which is a fancy ratio, basically, of saying how much is this microRNA expressed in lung cancer versus healthy people. And all of these 13 microRNAs were generally underexpressed in lung cancer, meaning that if you have lung cancer, you have less of this microRNA being produced in your body. By the way, microRNAs are molecules that regulate or reduce the expression of other proteins in your body or other genes in your body. So when you have less of a microRNA being made, the genes that it's supposed to control start going crazy. They start being produced too often. So some of these genes that the microRNAs are supposed to regulate include genes involved in fancy terms like the epithelial to mesenchymal transition and SMAD protein phosphorylation. I won't get into what all that means, but essentially these are all well-known lung cancer hallmarks. I also found that 6 of these 13 reliable microRNAs could reliably predict patient survival. So we generally found that people who had higher levels of these microRNAs tended to do better. I did a statistical analysis using these 13 microRNAs to try to separate lung cancer patients from non-lung cancer patients. The red dots are lung cancer patients, the green are healthy controls. As you can see, we're able to achieve a pretty good separation. It's not all the way there, but it's pretty strong. And here comes the AI, or machine learning. I was able to train some machine learning models to classify patients into lung cancer or not lung cancer based on the levels of these microRNAs in their blood. Now, interpretability is a big issue, right? We want to be able to interpret the results. So I also used a technique to basically ask the model, which of these microRNAs were the most helpful for you to make your decision about whether someone has cancer or not? And as you can see, a couple microRNAs, they're really useful, and other ones are less so. And so using that, I took the top #1 most useful microRNA. And using that alone, you're able to get about 80% accuracy, which isn't good enough for clinical use, but it's pretty good for just one microRNA. Now, if you use the top 2, you get to about 90%. And after that, you get diminishing returns. Using 13 microRNAs isn't really much more helpful than 2. And by using just a small amount of microRNAs in our model, it's more interpretable, it's more explainable, and it also costs less to run. So that's a brief, wrong button. So this is a confusion matrix which basically shows the model's correctness. The 1,154 and the 1,591 are the patients that it accurately classified. Excuse me. And the 145 and 106 are the ones it got wrong. So in general, it's very accurate. So I was able to identify a 2 microRNA combo that gets 90% accuracy and a 3 microRNA panel with almost 92% accuracy. So now on to drug repurposing. The pharmaceutical research process is really slow and costly. It costs about $1 billion to develop a new drug. I've seen estimates of $2 to $3 billion. And it takes 10 to 15 years to bring a drug to market because of FDA clinical trials and all that. So instead of having to develop a new drug from scratch, what if we find existing approved drugs and repurpose them towards new diseases? One of the famous examples is minoxidil, which was a hypertension drug, and they realized that all the guys they were giving it to started growing back their hair. So now it's used to treat hair loss. So I published this paper earlier this year on using machine learning-based virtual screening to repurpose drugs for a very common fungal enzyme, which causes an infection known as Candida albicans. I curated a data set of known inhibitors versus controls, so molecules that don't do anything for you. And I trained the machine learning model to predict the activity of these drug molecules. And finally, I ran that model on a database of FDA-approved drugs, and I found several drugs that could potentially be repurposed to treat this fungal infection. I'm wrapping up another project with my collaborators at UCSD, Dr. Tsigilny and Dr. Kuznetsova, on the same thing but for HIV. So now on to generative AI, or things you might have heard of are ChatGPT, DALL-E, Midjourney, all these new AI art tools. And I worked on a project a couple years ago, which is basically ChatGPT for proteins, or GPT-3 for proteins, in a very simple sense. So just to briefly explain what ChatGPT is, because I know that's on everyone's mind, it is essentially a really large machine learning model that has been trained on hundreds of billions of words. And through these words, it's learned patterns. For example, if I say, I had a great time visiting Naples, comma, it's going to predict the next word is probably Florida, maybe Italy, but it's not going to say Mozambique or Russia, right? Because those are not patterns that arise in English. So GPT-3 and ChatGPT are basically these huge models that have been trained to predict the next word over and over and over again. So I thought, why not do something similar for antiviral proteins to treat diseases like COVID or HIV? So I took some existing sequence data of peptides, which are just like letters. So in English, we have 26 letters in the alphabet. Long day, sorry. And in proteins, we have 20 amino acids, which are basically the letters of proteins. And so I trained a language model or a machine learning model to generate new peptides. I won't get into this graph, but I was able to generate realistic and potentially antiviral peptides. And they formed helices, which are these cool coils, which are known to be effective in helping fight viruses. Some other applications of AI include synthetic data generation, analysis of electronic health records and text data, enabling personalized medicine so you can have your very own diet plan, your very own cancer treatment, chemotherapy plan. But it's also important to be careful with AI. We've all seen Terminator. These things can be very, very dangerous. For instance, a research team was able to generate 40,000 potential chemical weapons, including VX, and it was relatively easy. I say easy, quote unquote, but it's not really that easy, but it was pretty easy. And all they did was they took a model that's supposed to help make drugs less toxic, and they reversed it. So instead of making less toxic drugs, it made more toxic drugs. They weren't even able to publish the full findings of their research report because some of the drugs they invented are potentially more deadly than anything we've ever seen on Earth. There's also other things to be worried about. For example, AI ethics and diversity of data. So for instance, the human reference genome, which we use often, is based on a Caucasian male, which may not accurately reflect women or Asian people or African people, people from around the world. And there's also issues with training bias if the data you're training your machine learning models are based on a specific subgroup. So in conclusion, AI revolutionizes how we do science. It can help us make better medicines faster and cheaper, which is good across the board for everybody. But we also must carefully assess the risks of what AI can and can't do and how it can do harm. So thanks again for inviting me to be here. I had a great time.